So now we'll look at how cells divide. This is called the cell cycle and mitosis. Mitosis is going to be responsible for asexual reproduction, growth and development, and repair. Mitosis is not how we produce eggs and sperm for sexual reproduction. That's called meiosis, and we'll get to that in part 3. So before we divide, the goal of mitosis is to make 2 new cells called daughter cells that are genetically identical. So we're going to start with one cell called the mother cell, and we need to copy the DNA so that when we divide into 2 cells, that each of the new cells has exactly the same DNA as the original cell. So the DNA is stored in the nucleus in long strands called chromosomes. Chromosomes consist of DNA wrapped around proteins. The complex of DNA wrapped around proteins is called a chromatin. A lot of the times we call it chromatin when it's loose, and then chromosomes when it condenses and becomes very dense for cell division. So these chromosomes have to be copied before the cells can divide. On the picture on the right, you can see this purple chromosome is a single copy of a chromosome. During interphase, the part before we actually start dividing, this chromosome is going to be duplicated. But it doesn't split apart, so here we're still going to call this one chromosome. Only now it's got 2 sides. These 2 sides are called sister chromatids. Eventually, when we get through cell division, these 2 sides will separate, and we'll have 2 separate chromosomes again, one for each of the new cells. So right here on the left, you can see a transmission electron micrograph image of a chromosome that has been copied. It's got a left side and a right side. These 2 sides are called sister chromatids. They are exactly the same, identical, because one was copied from the other. All right, the cell cycle is basically the life cycle of a cell. We're going to start right here with a brand new cell that just went through division. This cell is going to go through a long process called interphase before it actually divides, and then a short divisive process called the mitotic phase. Interphase is broken down into 3 parts: G1, S, and G2. G1 actually stands for gap 1, or first gap. But I like to think about it as growth. We named it gap because when the scientists were looking at this, originally we didn't have microscopes powerful enough to see what was happening in the cell. But what's happening here is the cell is just functioning normally, and it's growing larger. So you can see the size of the cell changes as it goes through G1. The S phase stands for DNA synthesis. Think about the word synthesis. What does that mean? Well, syn means to come together. Synthesis means to make, to create something. So DNA synthesis is where we're going to copy the DNA, like you just saw on the last slide. So this is where we're going to take one chromosome and copy it into a duplicated chromosome. And we're going to do that for all 46 human chromosomes. So by the end of the S phase, the cell has 2 entire sets of DNA. And then what we're going to do for the last part of interphase is called G2, or second gap. Again, this is a growth phase. What's happening here is the cell is making sure it has enough of all of its other components so that they can be split between 2 new cells. So it needs to have enough mitochondria, enough endoplasmic reticulum, so that when it divides, both of the new cells are going to have enough organelles. Once the cell has grown, divided or multiplied its DNA, and multiplied all the rest of its organelles, then it can enter the mitotic phase. The mitotic phase is here in blue, and this is the part of the phase where the cell is actually dividing. And this division phase is separated again into 2 more phases. the mitosis, M phase, and cytokinesis. Mitosis is specifically the division of the nucleus. It's only the division of the nucleus. Cytokinesis is the division of the rest of the cell. So cytokinesis is going to be the division of the plasma membrane and the cell organelles and all of the rest of the parts, the cytoplasm. You can have a cell that goes through mitosis and not cytokinesis, but usually they happen together. Usually cytokinesis happens in the very last phase of mitosis. So that's what we're going to see when we look at the phases of mitosis. Okay, so let's start looking at the phases of mitosis. On the top, you're going to see what it looks like under a light microscope, and then on the bottom, you see the Artist rendition. So this is interphase. This means that the cell is not dividing at this point. Inside of here, we have the nuclear envelope. It's fully intact. And we have the chromatin. See how it looks kind of like purple spaghetti? So the chromatin is very loose. It's not condensed. And what we see here is the centrosomes have duplicated. In that G2 phase, they get ready for division. So we have 2 centrosomes that are going to organize where our chromosomes are going to move around. Now we've entered into mitosis. Prophase is the first phase of mitosis. You can see in the light microscope image that you're starting to see squiggly lines here in the nucleus. Those are the chromosomes that are condensing and getting smaller and smaller. So you can see the same thing down here in the artist image. We still have a nucleus, and the chromosomes are starting to condense. They don't look like spaghetti anymore. You can clearly see the left side and the right side, the 2 sister chromatids. And those centrosomes have started making spindle fibers in between them, and they've started migrating to opposite sides. Prometaphase is the next phase of mitosis. Some textbooks will call this early prophase and late prophase. Your textbook takes prophase and splits it into prophase and prometaphase. So in the microscope image, you can see the chromosomes are even more condensed, but you can't see the nucleus surrounding them anymore. And these are the spindle fibers, the centrosomes, and they're on the opposite sides of the cell. So in the artist rendition, we see the same thing. These chromosomes have really condensed. They're much smaller, and you can clearly see the 2 sister chromatids. We have a lot of spindle fibers, starting from 2 centrosomes that are on opposite sides of the cell. And the nucleus is completely gone. All that's left are fragments as it is completely broken down. The next phase of mitosis is called metaphase. So if you see in the light microscope image, all of the chromosomes are kind of lined up here in the middle. Metaphase has one key characteristic, and that's that all of the centromeres, the centers of the chromosomes, are lined up here in the center of the cell. It's not necessarily right in the middle, more about right along the equator, right? Like if I took the cell and cut it out into 2 hemispheres, at this point where I cut it is where all of the chromosomes would be. The next phase of mitosis is called anaphase. Anaphase is associated with movement. So you can see in the light microscope image, these chromosomes have started to separate. They're not necessarily in the middle anymore. They're being pulled apart. So down here, these microtubules that are holding onto the chromosomes are getting shorter. The 2 sister chromatids were cut from each other at the center and are now being pulled apart towards the opposite sides of the cell. The last phase of mitosis is telophase. This image is also showing cytokinesis because they usually happen at the same time. But remember, cytokinesis is not technically part of mitosis. So in telophase, you can see that the chromosomes are starting to get looser again. They're getting longer. They're getting ready to be uncondensed. And the nucleus is starting to reform. These purple fragments of nucleus are reforming. We only have one centrosome, and the spindle fibers are all gone. Those yellow fibers are not here anymore. The cytokinesis that you can see is this cleavage furrow. This is an animal cell. So what it does is it invaginates in on all sides, and eventually it will get smaller and smaller and smaller and smaller until it pinches off into two separate cells. Cytokinesis is different in plants and animals. Animal cells develop a cleavage furrow. So that's this shallow groove on the surface of the cell. So this is a scanning electron microscope image. You can see the cleavage furrow kind of invaginating in. It kind of looks like a crevice. So what's really happening is there's a ring of microfilaments surrounding the center of the cell, and they just get smaller and smaller and smaller and tighter until it pinches the cell into two separate cells. Plants can't do this because they have a very hard cell wall, and so the cell wall can't just split into two. Instead, what happens is a cell plate, a new baby cell wall, starts forming in between the two halves. So these small vesicles will start to form along the midline of the cell. They eventually merge together to create what's called a cell plate. And then that cell plate will eventually reach the old cell walls and create a new cell wall, dividing both of these cells. These two cells are much smaller than the original cell. So now they have some room to grow before they have to divide again. But we don't want all of our cells to be dividing all of the time. If you think about the different parts of your body, things like your hair and nails and your stomach lining are constantly dividing. But cells like your heart cells and your neurons in your brain don't divide very much, and they usually stop once they reach adulthood. So we need to have mechanisms set up for controlling when cells are going to divide and when they are not. There are some that are called external factors. So external factors are things such as anchorage dependence, density-dependent inhibition, and growth factors. Let's start with anchorage dependence. So in the image on the left, you can see a petri dish where the scientists put a couple of cells on the petri dish. Because these cells are anchored to the bottom of the petri dish, they are going to grow. Same thing with the cells in the center. The cells in the center all are anchored. This is usually a requirement. That's why most of the cells in our body are attached to other things. Cells that don't require being anchored are usually called cancer cells, and they're growing unchecked. Density-dependent inhibition. So what does the word density mean? Density is the amount of stuff in a specific space. So if you look at the petri dish on the left, you can only fit so many cells in that area. Once all of the cells have filled up the area, what they're doing is they touch each other. And when they touch each other, that tells the neighboring cells that this area is full and they don't need to divide anymore. That inhibits cell division because of the density of cells in the space. If you remove some of the cells, that opens up a space. And so now the cells know that they have some room to divide some more. This would be like if you cut yourself and you injured your skin. So now those cells are going to divide to fill up that space. Cells also respond to something called growth factors. So in the center you can see an experiment where cultured cells are put in a nutrient liquid, one of them without growth factors and one of them with growth factors. The ones without growth factors fail to divide. The ones with growth factors divide and multiply and increase the presence of cells. Growth factors are really common in your body. When you cut yourself, the platelets that cause your scabs also release growth factors, which help to stimulate your tissues to repair. Those are all external factors. You can also have internal controls on the cell. These are called checkpoints. So in the picture on the right, you can see the cell cycle, and it's been made into a dial. There are 3 main checkpoints in the cell cycle, where we check the cell to make sure that it's doing everything properly so that it can divide properly. And if it's not, it stops the cell from dividing. The first one's called the G1 checkpoint. This one's going to be the most important. Most cells that pass this checkpoint go ahead to fully divide. The second one's the G2 checkpoint. This is our last minute check before division to make sure it has enough DNA and enough organelles and all of the DNA has been copied correctly. And this is the M checkpoint. The M checkpoint happens during metaphase, and it just checks to make sure that everything's lined up in the middle correctly and that the cell is going to divide in half correctly. Cells that don't regularly divide tend to be put into this category over here, G0. They don't pass the first checkpoint. And so things like mature heart cells are not constantly dividing. So they're not actually doing the cell cycle. This is really important to keep track of cell division, because when cells can grow without any regulation, they become cancer cells. So cancer is a really general term. All it means is unregulated cell growth. What happens with cancer cells is there's usually a mutation or some damage to the DNA, which causes a problem with the cell cycle control system. Maybe that cell can now release its own growth factors. Or maybe its cell cycle checkpoints have been messed up and it can't stop dividing when it's supposed to. So what happens when that happens is it divides into what's called a tumor. A benign tumor is a tumor that is going to stay in its original position. It doesn't move to anywhere else in the body. Given long enough, most cancers will continue to grow and will start to invade other tissue. So this tumor started on the gland. This is the breast, so this is the mammary tissue. And as it's dividing, it begins to invade the surrounding breast tissue. This is why cancer becomes such a problem, because these cells are growing so quickly that they start to outcompete the cells around them. And this is going to cause damage to the tissue, the healthy breast tissue, and can cause problems. A malignant tumor is a tumor that spreads in a process called metastasis. So metastasizing is when the tumor can break off and get into the blood or the lymph and spread to another part of the body. This is how you get something like colon cancer that eventually spreads and becomes lymphatic cancer. It's not because it's a new cancer, it's because cancer cells that started in one spot broke off, moved to the new spot, and then reformed and started making new tumors. Carcinogens are molecules or toxins in our environment that can cause cancer. So things like pesticides, UV radiation from the sun, some aerosols, radioactive chemicals, some preservatives in food, dietary fat, tobacco, and tar from smoking, certain paints, lead-based paints, asbestos that we used to use for insulation. Viruses can also cause cancer too. So herpes, hepatitis, HPV can all cause cancer. So we use vaccines to help prevent getting these viruses, which helps lower the incidence of cancer from them. In the last couple of years, the World Health Organization has actually updated its classification of carcinogens to include meat. So a class 1 carcinogen, this first bubble up here, is a scientifically shown to have a causal relationship with cancer. It means that these things cause cancer. So in this box, we have processed meats, including sausages and hot dogs, bacon and salami. these are just as bad as these other carcinogens up here. So tobacco smoke, asbestos, the HPV virus, those are all also in this group 1 carcinogens. Group 2A is shown that it probably causes cancer. There's a high correlation between eating these and getting cancers. So they've included in these red meats from pigs, beef, and lamb. Think about your diet and think about how much of these carcinogens you regularly consume on a daily basis.